Ladies and gentlemen, welcome to the vaccine presentation and thank you for your interest. And thank you to the organizers for the opportunity to present here at BioKorea. Our company, Vaximm, is developing oral immunotherapies for the treatment of cancer. My name is Heinz Lubenau. I'm the CEO and co-founder of the company. What we are developing is an oral DNA vaccination platform, which is very flexible, powerful, and robust. It's a true plug-and-play platform technology for the oral delivery of DNA. It consists of two components. One is the live bacterial carrier, which is a prophylactic typhoid fever vaccine, which has been on the market for several decades and has been administered in more than 200 million doses, so it's known to be safe and well tolerated. We do not touch the bacterium itself, but we transform it so that it becomes a genetically modified organism and carries a eukaryotic expression plasmid. And with the DNA of this expression plasmid, we can encode whatever target antigen we want to encode, against which we want to raise T cells, which are systemic and target specific. So it can be a large protein, like in case of our first product, Vaximm Ag, where we target the VEGFR2 receptor consisting of 1,350 amino acids, but we can also encode small peptides or a series of small peptides like a string of beads. With our lead program, Vaximm Ag, we are in phase 2 clinical development for recurrent glioblastoma, a very hard to treat indication, where we have seen promising results, including monotherapy activity, but also activity in combination with the checkpoint inhibitors. We have a clinical trial collaboration in place with Merck and Pfizer, which covers two indications, recurrent glioblastoma and metastatic colorectal cancer. And Merck and Pfizer deliver to us the anti-PD-L1 checkpoint inhibitor avelumab for free. In addition, we have a very exciting personalized neoantigen program. We think that our platform has clearly differentiating features with which we may overcome many hurdles which cannot be overcome by competing neoantigen targeting technologies. We have established a first partnership with the Japanese company NEC. NEC has an artificial intelligence platform. They select and identify the mutated antigens, the neoantigens. We have the delivery platform to bring it into the patient as an investigational medicinal product. We are planning to start our clinical trial in summer this year. We are a private company founded in Switzerland with a subsidiary in Germany. The company was founded as a spin-off from the German Merck and BB Biotech Ventures, and the technology was initially invented by the Scripps Research Institute in La Jolla in California. We have three collaborations in place, one with Merck and Pfizer. They provide to us the avelumab, the anti-PD-L1 checkpoint inhibitor for free. NEC, we collaborate on our first personalized neoantigen targeting trial. And China Medical Systems, they are our commercial partner for China and other Asian countries, including Korea, for all the vaccine products. So the investors are mainly from Europe and from Asia. It's BB Biotech Ventures, BCM Europe, Biomed Partners, China Medical Systems, M Ventures, which is the venture arm of the German Merck, NEC, and Sunstone Capital from Denmark. This slide shows you how our system works. For the patient, it's very easy, it's very efficient. So the patient just drinks 100 milliliters of water, where the live bacteria, which carry the eukaryotic expression plasmid, are suspended. We add some buffer salts so that the live bacteria survive the acid medium of the stomach and reach the small intestine. And there in the small intestine, they are recognized as foreign bacteria, and the human body wants to get rid of these bacteria. So what happens is that the bacteria carrying the plasmid are taken into the so-called Peyer's patches, which are part of the lymphatic system of the gut. The bacteria are taken up by the macrophages, the eating cells. The bacteria die, and the plasmid is released, goes to the nucleus, and initiates an antigen expression according to what we have encoded at the very beginning. So we use the human body in a way that the human body is our production facility for the antigens which have been encoded in our DNA. The macrophages, which produce the antigen, they start to die, they get apoptotic. The apoptotic vesicles are taken up by the dendritic cells, and the dendritic cells present the processed protein or the peptides to the immune system. So that's the second function of the human body. The human immune system presents the antigen, and what the result is that we have a systemic antigen-specific T cell activation targeting the antigen which we have encoded at the very beginning. So the T cells which we generate, they are antigen specific, systemic, travel around in the bloodstream. They can cross the blood-brain barrier. That's important for our glioblastoma project. Detect the presented antigens presented by the tumor cells, expand there and destroy the cells. That's how the system works. A very efficient and a very robust system. In our first product, Vaximm 01, we have shown the platform clinical proof of concept. We have treated in the meantime 91 patients in 3 different cancer indications, in pancreatic cancer, in colorectal cancer, and in recurrent glioblastoma. And in recurrent glioblastoma, we have seen the most promising and the most encouraging results, including clinical responses, meaning partial responses, and 1 complete response in that very hard to treat indication. Partly, these results were generated as monotherapy, under monotherapy with Vaximm 01, but also in combination with a checkpoint inhibitor. In the first 12 patients which were not resected, which presented with a growing tumor, which were not resected, and which were treated with Vaximm 01 in combination with a checkpoint inhibitor, in 2 different trials, we saw 5 clinical responses out of the 12 patients treated. On the right side, you see the 3 patients which were treated in the first trial, in our exploratory monotherapy trial, where the doctor added nivolumab after 6 weeks or 8 weeks of treatment, and 2 out of 3 patients had either a partial response or a complete response, and the complete response had already developed to a partial response under monotherapy. These data were very encouraging and intriguing. So we agreed on the clinical collaboration with Merck and Pfizer with the provision of the anti-PD-L1 checkpoint inhibitor avelumab. And again, in the first 9 patients, we could confirm what we saw in the first trial. So again, 3 partial responses, so that we have out of the first 12 patients, 5 which had a clinical response, corresponding to an objective response rate of 41.6%. And these 41.6%, which are highlighted here on the upper part of the slide, are clearly higher than what we see with Avastin monotherapy. Avastin has been approved in the US and in Japan, but not in Europe. What we see with Avastin monotherapy is an objective response rate of around 22-23%. With checkpoint inhibitor alone, we only have an objective response rate of 7 or 8%, so clearly below 10%. And what we are going to do now is we extend the trial and hope to be able to confirm what we see in the first 9 patients plus the 3 from the first trial. We are now targeting personalized neoantigens as our next and very exciting project. From our point of view, we have one of the very rare, if not the only commercially viable neoantigen platform. On the one hand, in terms of pricing, it means in terms of manufacturing costs. And on the other hand, and that's a much more important argument, time to administration. So as we have a live bacterium, we do not have to do a sterility test. The bacterium carrying the plasmid which encodes the neoantigen is the product. So we have a very lean, very fast manufacturing process, and we have a much faster turnaround time than other approaches, as we, for example, do not have to do a sterility test, which always takes 3 to 4 weeks. We can target a large number of neoantigens of any size. We can encode 9 MERS, we can encode 25 MERS. We have a validated delivery technology through our first product, Vaximm 01. We have shown clinical responses, we have shown immune responses, we have the manufacturing process in place, which is very straightforward, a 1-liter bacterial fermentation, which delivers enough material to treat a patient for more than 2 years. And the manufacturing costs are very small, as we have just a 1-liter bacterial fermentation, and we still have a margin to make money if you take into account that the reimbursement per personalized treatment in the US is estimated to be between 70 and 150,000 US dollars. We have already established a first strategic partnership with NEC and want to go into the clinic in summer. We want to treat patients in cancer indications with a high unmet medical need, and we can combine directly with immunotherapies. And our approach is applicable to other T-cell approaches, including personalized or shared mutated tumor-specific neoantigens. What we want to do in our first clinical trial together with NEC is we want to do a signal finding study, a basket trial, where we start with a small number of patients, where we want to detect signals with regard to safety, immune response, biomarkers, and efficacy. And we want to test 2 doses, 10 to the 7th and 10 to the 8th. And the Data Safety Monitoring Board will be involved in the dose escalation. We want to directly combine with the checkpoint inhibitor in patients who are under checkpoint inhibitor treatment, standard of care therapy, and have experienced stable disease or partial response. Patients will be treated for 6 months, including an initiation treatment, 4 doses in 1 week, and then 4 weekly boostings up to week 24. So in summary, we are seeking to raise 15 million euros. We have a very powerful and highly flexible plug-and-play platform technology for the oral delivery. We have a very good safety profile, and the manufacturing costs are very, very reasonable. With our lead program, we have very promising results in recurrent glioblastoma, one of the toughest indications, including single-agent activity. We want to extend the program to other indications, including colorectal cancer, and we see clear synergies with checkpoint inhibitors. And our market potential has significantly increased by our China partnership with China Medical Systems. In addition, we have our very exciting personalized neoantigen program, where we see clear differentiating features with which we can overcome major limitations of other neoantigen approaches. We have a very short time to administration. We want to get it done from the list of neoantigens to the oral administrations within 15 days. And we have affordable manufacturing costs. So we can make this neoantigen treatment, which is personalized, affordable, and available at short notice. And we have established a first partnership with NEC, and our first clinical trial is planned in summer this year. Thank you very much for your attention, and I'm available through the partnering system for any question you may have. Thank you very much.